we move on to the next speaker, who is Dr. Suhas Sureshchandra from the University of California, Irvine, and he will talk about tissue determinants of the human T cell receptor repertoire. Just briefly to introduce him, he received his Ph.D. at University of California, Irvine, where he investigated fetal immune consequences of early life stress exposures, such as high-fat diet and maternal obesity. And since 2021, he's a postdoc there, studying the diversity of T follicular helper cells in various populations. And he's broadly interested in applying systems biology to B and T cell responses and vaccines and viruses. And very much looking forward to your talk.
All right. Thank you, Victor and Pam, for organizing the seminar series and letting me present some of the data that we've generated in the lab recently. I'm a postdoc in Lisa Wagar's lab, and today I'm going to be talking about switching gears from B cells and talking a little bit about T cells. Okay, so since the pandemic, there's been renewed interest in understanding human tissue immunity. And the tissues include lymphoid tissues like lymph nodes and spleen, or mucosal tissues like lung. But what's interesting is that these tissues, they harbor unique immune cells. So immune cells that are very rarely detected in circulation, sometimes detected in circulation post-vaccination or infection, but under homeostasis, these are quite restricted to tissue compartments. So this includes CD8 resident memory T cells. Within the CD4 compartment, you have the T follicular helper cells, and within the B cells, you have the germinal center B cells, as well as long-lived plasma cells. What we also realize is that the immune responses actually occur in tissue sites, and what you're seeing in blood is just a sampling of different tissue sites. So this commentary from Donna Farber sort of describes why there is an urgent need to sort of look at tissue-specific responses, particularly if you are interested in looking at humoral responses to vaccines or viruses. But given the logistical issues with accessing tissues, as well as ethical issues of sampling cells from the human tissues, blood has always been the surrogate for most of the analysis. And blood is great. It gives you a snapshot of what's happening in the tissue. But if you actually look at the immune cell distribution over different compartments in the body, so here each tiny square represents roughly 1 billion cells in the human body. And in maroon, what we have are mostly the T cells. So the T cells are mostly restricted to lymphatic system, lung, skin, or the GI tract. And what you're seeing in blood is roughly just 2% of the total T cells in the human body. What we know about human T cell receptor repertoire is coming from 1 million cells, roughly 1 million cells that you've sampled from blood. So which is great, gives you a snapshot of different T cells from the body. But there is this whole black box of T cells that reside in tissue that are phenotypically never found in circulation. And we don't know much about what's common between blood and tissue. So certain questions that are still pending, and one of the open-ended questions is how many clonotypes are there in the human body? Can we even have an estimate of experimentally estimate the total number of clonotypes in the body? And are the clones that you're sampling in blood, how well mixed are they with what you see in tissue? So the central question for the talk would be, what do we miss by only profiling blood in humans? So to address this question, we performed a very simple but very expensive experiment where we profiled paired blood and tonsils, so T cells from paired blood and tonsils from 10 donors. So we focused on few donors because we wanted to enrich and look at a large number of cells. The goal was to look at least 1 million cells from each of these donors. So 500,000 from blood and 500,000 from tonsils. And we're able to sort out the alpha beta positive T cells and then profile the cells using single cell RNA sequencing. So we use the 10x platform with paired VDJ sequencing to look at the TCRs. The cohort basically has 10 donors across age spans, so starting 3 years old all the way up to 30. And then we have a mixed set of immune histories across these. So at least with the herpes viruses, you're seeing mostly naive donors, but also some of them who are exposed to both. And to sort of assist with the analysis, we used a secondary cohort of donors where we shallow sequenced the cells, but we were able to sort of tag them with surface markers, so oligotagged surface markers, so we can look at the protein information in these clusters as well. So this is just a cartoon of the analysis, very basic analysis using 10x, where we were able to sort of remove B cell contaminants from mostly tonsils, because tonsils roughly are 60% B cells. And then within each donor, we were able to sort of integrate the blood and tonsils to get a composite UMAP for that particular donor. And then we were able to integrate all of those donors with the protein data from the cohort of donors with the protein data using the atomic sketch integration, because this was roughly millions of cells being analyzed here right now. And then that gives us the total UMAP of cells from blood and tonsils. And even though the expectation was to get 500,000 cells from each of those compartments, we know from single cell sequencing, the number of cells sort of like drop dramatically. So this is depending on the quality of the sample as well as the QCs that we were performed. So for most of the donors, we are able to see roughly 60 to 80% of the cells having a productive TCR. And this is distribution of the number of cells in blood or tonsils from each of those donors. So we didn't get 10 million cells as we expected, as we had planned, but we got roughly 5.7 million cells. The first question we asked is, given the number of cells that were sampled in this data set, are we able to even see a saturation of the number of clonotypes? As expected within each of those donors here, as you sample more and more of the cells, you're able to see more and more of the clones. But what was interesting is that if you actually downsample, you're able to sort of see the error rates in which you falsely call a cell as a singleton. So when you sample high enough, you're able to see these expanded clones with very low error rates. But as you downsample, and this box basically tells you the range of number of cells that are basically sequenced in most routine single cell RNA analysis in human T cells, most of the cells that you would have falsely called as singletons are actually expanded clones. So this sort of gives you an idea of why oversampling is kind of important to sort of make this true estimate of expanded clones. So here's a UMAP of, this is almost a year's worth of analysis to get to the point of data collection from all the 10 donors and collect 5.7 million cells, and then integrate and annotate all of these clusters. And most T cell aficionados will recognize these subsets, but for those of you new to see T cells as naive cells, there's memory subsets of central memory as well as effector memory, and a couple of these RNA markers that were used to sort of distinguish different subsets. And I've used protein markers as well. There's T follicular helper cells and a subset of T follicular helper cells that express high levels of IL-21 and CXCL13. And then within the CD8 compartment, there is the resident memory T cells and mates. So the goal of the project was not to sort of build an atlas of transcriptomic diversity of T cells in tonsils. There's already a tonsil atlas that actually does that. But our goal was to sort of have a confident sort of annotations to make correlations between the TCR and phenotype. As you can see, if you split the data set by compartment, you're able to see there's a big chunk of cells that are not present in tonsil, not present in blood, but present in tonsil. These are mostly your T follicular helper cells and a lot of the resident memory subsets. And at the bottom, you're able to see the distribution of those cell types. So in tissue compartment on the right here, you're able to see there are fewer naive cells, so green and yellow, but there's more of the central memory cells as well as these unique clusters of T follicular helper cells. There's more T regulatory T cells in tissue as well as the TRMs, but there's more mates and sort of effector memory subsets in blood. But within the clusters that exist in both compartments, that is blood and tonsil, there's differences in distribution of the clone sizes. So effector memory cells are more clonal in circulation, whereas central memory cells are more clonal in tonsils. Okay, so there's differences in the phenotype, there's differences in their clonal distribution, but what about the mixing of the two compartments? So here, for these 8 donors, each of those dots is a specific amino acid sequence that is a clone. And then on the x-axis, we're looking at the size of that clone in blood, and y-axis, you're looking at the size of the clone in tonsils. So as you can see, the circles in red, they're basically talking about depicting the clones that are shared between the two compartments. And the frequencies are rather lower than previous estimates. And if you actually look at the extent of mixing, there's an age-dependent increase. So roughly the extent of mixing is 1 to 7%, and there is an age or exposure associated increase in this functional overlap between the two compartments. If you focus on the clones that are actually exclusively present in tonsils, so these clones that are large in size but have no representative in blood, these are mostly the T follicular helper cells. So when you sample blood, you are less likely to find a lot of these clones that are present only in tissue. So what about the antigen-specific compartment? So what we've seen so far is a poor mixing within the total T cell compartment. So to focus on the antigen-specific cells, we identified 4 donors who are HLA-O2-O1 allele, and we depleted CD4s and CD19s and just focused on the CD8s. And then we used the DNA bar-coded tetramers to isolate antigen-specific cells. So we focused on a couple of naive antigens as well as viruses with prior exposure. So you have EBV, CMV, SARS-CoV-2, and flu, a total of 15 peptides in this multiplexed tetramer experiment. We sorted out the tetramer positive cells, and using the barcode, we were able to identify the specificity of these cells, but we also have the gene expression, protein, and matched TCR for each of those cells. The UMAP of these cells, you can see these, there are distinct phenotypes of antigen-specific cells. There is phenotypes that are dictated by compartments. So in blood, these antigen-specific cells are more likely to be MRAS or CD95 bright cells, or to some extent Th17-like cells. There's naive clusters in both blood and tonsil, but in tonsil, the antigen-specific cells are more likely to be either a few of the central memory, but mostly resident memory T cells, which are never found in circulation. If you look at the distribution of those antigen-specific cells, and this is a composite of all 4 donors, with CMV, you're able to see most of the cells are restricted in blood. In flu, it's a mix, but with EBV, depending on the peptide, we're able to see this distribution of BMLF-specific cells, which are more likely to be found in blood, versus EBNA-specific cells that are more likely to be found in tissue. And within the naive donors, most of the cells are as expected in the naive compartment. So suggesting that even within a virus, you could probably have immunodominance might be dictating the location of those antigen-specific cells. So not just the location, the clonality is also quite different. So the BMLF-specific cells that are found mostly in blood, they do have representatives in tonsil. As you can see, the size of the clone might be quite distinct. So this is clonal tracking analysis, focusing on the largest, the top 10 clones in blood on the left side, and tonsil on the right side. And these are for 2 different peptides from the same virus. But for EBNA-specific cells, the largest clones in either compartment do not have representatives in either blood or tonsil. So this suggests that there's limited sharing of the antigen-specific clones between blood and tonsil. So the next question we asked is, how is the phenotype linked to TCR? So within a compartment, if you have 2 randomly selected TCRs, identical TCRs, how likely are they to have the same phenotype? So the answer to the question is depending on the level of annotation. So for CD4s and CD8s, more likely the relationship is quite conserved. So if you take 2 TCRs of similar identity, they're more likely to be the same cell, CD4 or CD8. And as you go deeper into annotation as naive memory, this relationship is still pretty tight. But as you go into memory subsets, so central memory versus effector memory or regulatory T cells, or into granular subsets of T cells based on either expression of certain chemokine receptor or integrins, this relationship sort of weakens. So this sort of makes sense depending on the plasticity of cells. So CD4 to CD8 determination happens early on. Naive to memory, depending on the exposure, is a unidirectional transition, versus memory subsets or granular subsets, you see more plasticity. So you see this weak relationship between TCR and phenotype. So what about cell types that are restricted to tonsils alone? Are we able to see any clonal relationships for those cells in circulation? We focused on CD8 resident memory cells. So there's large proportions of those cells present in tonsil, but very rarely observed CD8 resident memory-like cells, at least transcriptionally seen in circulation. So we're able to see these cells because we sampled millions of cells from blood from each of those donors. So these cells make up roughly 1.4% of the memory CD8 repertoire in blood, but they're related to roughly 10% of the repertoire in tonsils. So there's a small subset of CD8 resident memory-like cells in circulation that are clonally related to roughly 10% of memory CD8s, and they're mostly CD8 resident memory cells. So there's tight clonal relationship between the two subsets in circulation and in tonsils. So this could be your XTRMs that have been recently described. These are TRMs that exit the tissue and they're able to sort of move into other compartments and establish residence, or could be cells that are XTRMs that differentiate into other effector subtypes in circulation. T follicular helper cells, these are abundant in tonsil, but not so much in blood. But roughly 1.3% of tonsil CD4s or CD memory repertoire are clonally related to less than 1% of the repertoire in blood. So this is the opposite way of looking at it. So you have roughly 1% of cells in tissue that are clonally related to 1% of the memory's repertoire in blood, and they are more likely to be central memory or effector memory cells, but with specific homing markers. So these could be, again, cells that have recently exited, or could be TFH that sort of transit from one second lymphoid organ to another. So what we've seen so far is all of the T cells directly ex vivo. We're trying to make relationships between the TCR as well as the phenotype. But what about cells that sort of evolve over the course of antigenic stimulation? So our lab has established the immune organoid system where we sort of aggregate your tonsillar cells into organoids over the course of 2 weeks, stimulate them with antigens. So this is all ex vivo vaccination, in vitro vaccination, and then the organoids reaggregate, after which we're able to look at T and B cell responses, specifically to flu. So on day 7, 10, and 14, I sorted out the activated T cells. So this is 38 positive, HLA-DR positive, CD4s and CD8s. So these cells were sorted out and sequenced using the 10x platform with TCR. So the goal was to look at specific clusters of T cells that sort of emerge in the stimulated conditions, and then use a TCR as a barcode to track the cell's phenotype on day 7, 10, and 14 compared to where they began. So this is a UMAP of all of the activated cells, and in the interest of time, I'm just going to focus on the CD8 compartment. So there's 3 clusters: the resident memory T cells, 41BB expressing CD8 T cells, as well as proliferating CD8 T cells. And if you do a clonal analysis, on the left side, I'm focusing on just the new clusters that emerged within the activated population in organoids. And on the right, I have what their phenotypes in either blood or tonsils are directly ex vivo. So CD8 resident memory cells are the ones that are proliferating highly. They're clonally related to the proliferating CD8 population. But as you can see, they do have, they're clonally related to either resident memory or effector memory cells in tonsils with little representation in blood. But the 41BB expressing cells, these cells are again responding to stimulation, but you don't see clonal relatives of these cells in either blood or tonsil, suggestive they could be coming from the naive compartment. So you have 2 pools of phenotypically distinct antigen responsive cells that have quite distinct clonal origins as well as the phenotype. Okay, so the paper is out on bioRxiv, and there's a lot of analysis, and we're still working on some of the revisions. But in the take-home message is that there is good phenotypic mixing between the 2 compartments, but not clonal mixing, at least based on estimates from Donna Farber's group. The numbers that we are seeing are much, much lower. This is primarily because I think phenotypically CD4s are less mixed compared to CD8s. We also sort of demonstrated to some extent that the tissue site might be dictating immunodominant responses. And then you don't see, even though you don't see phenotypically some of the cell types that are probably quite important for responses to vaccines or viruses, resident memory cells or TFH, you do see their clonal relatives in circulation. And that sort of leads us to some understanding of the plasticity of these cells, at least, which has never been shown in humans. And I think this has broad implications for T cell profiling, particularly post-vaccination. And there's technology that's moving further towards sequencing millions of cells. Maybe not with their actual phenotypes, but at least if you can do an alpha and beta sequencing, alpha and beta TCR chain sequencing for millions of cells, that might be more relevant than just focusing on like a couple of thousands of cells to make implications on clonal expansions. With that, I'll thank the lab. This has been a massive effort from Naresha's Aligramas lab in WashU. Alyssa and Elizabeth were the ones who did all the sorts and the sequencing. And James from Andreas's lab has helped me with the TCR analysis, and a lot of people in the Wagar lab have helped me. I'd like to thank the tonsillectomy patients and their families and the clinicians who've helped with tissue acquisition and funding sources, particularly Welcome Leap for providing us funds for this massive endeavor. And with that, I'll take questions, but also have my contact information if someone wants to reach out and talk about the data set.
Thanks so much. Amazing data set. Are there any questions? If not, I can maybe start with the question. So, I mean, I thought it was really compelling that you showed that there was this poor mixing between the different compartments of the clonotypes. I think this we cannot stress this enough that this is actually the case. So only profiling the blood is not sufficient. Especially, I think this is the case for diagnostic approaches where you only focus on the blood, but actually the tissue sites is where the pathology might happen. So yeah. So you mentioned also the antigen-specific part with the beam, and I'm asking then the same question as I asked to Scott. So for the EBV, etc., TCRs, did you find some overlap with public databases, or were these completely novel clonotypes that you
Not as many cells as we would have expected. At least with the naive, they were all novel, but with the memory compartment, there were a few hits from VDJDB, and that's I think that's where we're going, is trying to sort of look at what percentage of those cells are actually validated and being shown to be in O2O1 donors. Yeah. So it's not as high as, not all of them are on the database.
Yeah. Do we have more questions for Suhas? I'm sure people have questions.
Also, with that analysis, I focused at least for this study, I focused primarily on cells with confident single specificity. There is roughly 20% of cells with multiple specificity, and that's challenging to know. They are higher than the background, but it's challenging to know what that necessarily means in terms of like.
Yeah. Yeah, I think there's a paper from Morten Nielsen also where he published a method of cleaning up these kind of data sets to really be sure to know that what one is looking at is really specific TCRs and not some cross-reactive or something. Yeah. Nina, do you have a question?
Well, a couple comments actually. It's really great that you did this with single cells, and I think one of the things, Donna and I were working on this with bulk sequencing, which has some advantages in terms of sampling. So I think there you do have to be a little bit careful about claims of lack of overlap specifically, but with single cells, you can have much more confidence that the clones that you're calling overlapping are truly related to each other because you have the paired chain data. So that's fantastic. And then being able to couple that with some of the antigen specificity data is also really, really nice. I think we also saw a hierarchy of overlap. In a paper that we published in Genome Medicine that might be relevant to these observations as well. So
yeah. You're talking about the Miran et al.
Yes, with Michel Miron, there was a maintenance of human memory T cell repertoires. And yeah, so we saw that the CD8s overlapped extensively, especially the TAMRAs across different tissues. But some of the CD4 subsets can overlap as well. It's just they tend to be a lot smaller clone sizes and much more diverse. So it's much harder to show the overlap. And then there's also a hierarchy, as you mentioned, in terms of lineage and subset. And there is a plasticity by subset for sure. Yeah. So all those things I think are very consistent with what we saw.
Do you have plans also, or someone in your lab to expand this to B cells to also have such a huge data set for B cells?
Yeah, so the goal was to have a parallel 10 million B cell project from the same 10 donors. So that was the goal. But as you know, there's not a lot of B cells in circulation. At least when you save one tube of PBMCs, you don't get many B cells. So we're we think we're dealing with roughly maybe 3 million B cells, and that study is gonna probably come out next year. Sorry.
Oh, very nice. Very nice.
The goal would be to sort of characterize the B cell phenotypes, their repertoire within those 10 donors. We do have organoids from those donors as well, so you can actually look at flu responses. And for people who build trees, you need a lot of germline cells, so this is a good resource for that. But also we're interested in like the T cell B cell interactions. That I think is the focus, because you have the GC-B cells, you have the GC-TFH, and that's the unique aspect of this data set that gives you power to look at those interactions.
Exactly.
And my interest is definitely in the aging side, so I'm interested in how early life TB interactions vary compared to adults. Yeah.
Great. I'm looking forward to the data set. And then, as you said, there are too few data sets where we have both in one B and T cell, so we can look at interaction. Yeah, this is great. And so are there more questions? If not, I think we can close here. No? Okay. So then we meet all again, hopefully, I think November 21 for another AIRR-C seminar. Thanks again to both of the speakers. Really nice talks. And both of the talks will be on the YouTube channel, hopefully soon. Thanks, everyone. Have a good evening or rest of the day. Thanks, God.
Take care.
Bye-bye.